Hi students. Today I would like to talk about a very important topic, a topic which is of much importance to the society as well as to the forming of a healthy family. We might have come across different different offsprings in different families, which are the light in the lamp of their family. And quite often happy family is always around the happiness of the child. But if a child is having any defect or any birth defect or any even a small cough and a cold, what happens? That would result in the unhappiness of the family. So coming to a small, leaving a common cold or let it be a fever, leave all that aside. But if a child is having a newborn infant is having a birth defect, that of course adds, of course, though, however, we say that we would take care of the child or what. It is a crisis for the child as well as for the family, and they have to overcome that in a in a very patient and a tactic way. So now, what is the reason behind this? Okay, that's what we're going to analyze on the basis of mutations. We're going to say in US, they say that every 4.5 minutes, a baby is being born with a birth defect. And one in 33 of the babies in each year are having a birth defect. Okay, so now why do I have to say about this in US but in India? Okay, not in India. Okay, of course, the statistical evaluations which have been available is not that much here. So that's why I took US as an example here. So if you go to consider these birth defects which are prevalent among, say, there are various reasons which are responsible for it. It could be some environmental factors. It could be any nutritional parameters which would result in the, like if you don't have folic acid and all that in the sufficient level, it could live into birth defects. Or it could be some other, okay, in environmental factors itself, we can have lots of like the occurrence of certain viruses. These all could contribute to birth defects. Another contribution is being given by the genotypic factors. And these genotypic factors are mainly because of some mutations which could happen within the what? Within the replications, DNA replication, or within the either in the egg formation or in the sperm formation, or after the zygote has been formed. In all these stages, when some mutations happen into the genes, they could also contribute to the birth defects. And we had told about the different mutations which have been possible. We had told about we had told about the gene mutations, that is, keep it aside. Now coming to chromosomal mutations, they are mostly being found to be associated with these birth defects. And we found earlier about the polyploid condition, which has been present in the case of plants and animals. But coming to birth defects in humans, most of them is being found to be associated with a condition called aneuploidy. Now, what is aneuploidy? Aneuploidy is a situation where additions or deletions to the diploid chromosome number happens. That is, where you know the diploid chromosome number. The diploid chromosome number is 2n. When this diploid chromosome number, an additional chromosome is being added, or it can be not one single chromosome, it can be any number of chromosomes, or when some chromosomes are being removed from it, that will result to a condition called aneuploidy. Aneuploidy, ploidy, I told, it's a chromosome number. So when there is a change in the chromosome number by adding or deleting some chromosomes to the diploid chromosome number, that could lead to the aneuploidy in the case of humans. And this mainly, it occurs as a result of non-disjunction of the chromosomes during mitosis and meiosis. So what happens? The offspring will either get an extra or they might lose a chromosome. So, and the naming of the aneuploid condition is generally based upon the number of chromosomes that have been added or deleted. Now, if you have a deletion of about one chromosome, you consider it as a monosomic condition, or sometimes you might have an additional one, so you call it as a trisomic condition, or you might have lost two chromosomes and you call it as a nullisomic condition, or sometimes you might have n plus one, that is a disomic condition. Like this, different terms have been given to this aneuploidy conditions. Now we told about disjunction and non-disjunction, and we told that a failure in the non-disjunction during mitosis and meiosis is responsible for aneuploidy. So let me just look what is non-disjunction. See, now during the normal mitosis that we have, you have the parent cell which is having two chromosomes here, and during DNA replication, the homologous chromosome is also being formed, and mitosis happens, and the chromosomes get split to form different chromosomes. That is, the homologous chromosome will split and you will get different chromatids. And further on, what happens? See, similar to the parent cell, you get a daughter cell which is having the 2N configuration itself here. 2N genome orientation is also being formed in a normal mitosis. But sometimes so you just remember, you are cutting here during the process of mitosis. At the kinetochore, the chromosome gets split, okay, and it will result in the formation of daughter chromosomes here. Now, sometimes in certain conditions during mitosis, what happens is when the proper disjunction will not happen, that situation is called as a non-disjunction. What happens? Some of the cells might get probably three of these, three chromosome, daughter chromosomes, and one would get, now for example here, now the original one was supposed to be one blue and one red here. It got an extra blue, so this is referred to as a trisomic condition, that is 2n plus 1. Okay, here it is losing 1. Okay, so instead of getting one blue and one red, it has lost a blue here. So such a situation is also called a aneuploidy. Now coming to meiosis. In the same way, during meiosis, what happens? The homologous chromosomes will have to be what? You see, during the process of meiosis, during a proper disjunction, what happens? The daughter chromosomes are being split together. No, the daughter chromosomes are being formed by splitting at the kinetochore region. But sometimes you can see, due to non-disjunction, what happens? The whole chromosomes will be split and be carried into one. So the proper formation of the mitotic spindle, or the proper functioning or splitting of these homologous chromosomes will not happen here, and that would probably lead to non-disjunction, and it would lead to a situation of an extra chromosome to be added to one cell or an extra chromosome to be lost to another cell. So that is referred to as aneuploidy. Now, as I told you, there are different terms by which you call it. If you lose a chromosome, it is referred to as monosomic. And if you have an extra chromosome, now if you lose two chromosomes, it is referred to as nullisomic. And if you are having extra chromosomes, like if you have an extra chromosome 1, it is referred to as a trisomic condition. Or if you have a double chromosome, okay, then it is referred to as a 2n plus 1 plus 1. That is referred to as a double trisomic. Now you just have a look, what is this here, and you might think 2n plus 2, that is tetrasomic. What is the difference between these two? I'll be telling you. 2n plus 3, that is referred to as pentasomic. Now just look at this situation. 2n minus 1. If this is a normal chromosome level, okay, AA, VB and CC. These are the homologous chromosomes in 2n. It's called to be 2n because you have two copies of A. You have two copies of B, you have two copies of C. That is a diploid chromosome. For each chromosome, there is one more pair. So that is a 2n condition. Okay. Now, in one case, what happens? You are losing a C over here. That is, you are losing a chromosome C. So that becomes monosomic, 2n minus 1. And 2n minus 2 here. both the C's are being lost. So you become 2n minus 2 or the nullisomic condition. Now, and here you have AA, BB and triple C. So you have an additional C here. That is 2n plus 1 trisomic. And in this case, you can see that AA, BBB and CCC. So you have an extra B as well as you have an extra C. That's the reason why you write it as 2n plus 1 because two different chromosomes are being increased here. Then tetrasomic here, but here it is, what does it mean? One chromosome itself, it is being duplicated twice. So that's why you write it as 2n plus 2. And here in pentasomic, you can see that one chromosome itself is being, rather than having two C's, we have five C's. So it becomes 2n plus 3 pentasomic. Now this could lead to different diseases among humans. Okay. Now in the case of Turner's syndrome, which has been found, you will one chromosome, one X chromosome is missing. And nullisomic is a highly inviable stage, and normally the fetus will not survive such a nullisomic condition. And Down syndrome, which has been found in, which is very quite common, it is one of the most predominant birth defect which has been found among the human. That's because of a trisomic condition, as well as a second prevalent one is a Klinefelter syndrome, which is also a trisomy. Then you have Edwards syndrome as a double trisomic example, and mosaicism in tetrasomic. You will be seeing some figures about that. And then you have the murine mammary tumorigenesis if there is a pentasomic condition in the 13th chromosome. And then you also have a penta X chromosome defect in the case of humans. Coming to Klinefelter's, okay, the most prevalent one, we'll start from this. Down syndrome, what happens in the 21st pair of the autosomal chromosome? Okay, you have autosomal chromosomes as well as a sex chromosomes. In the 21st pair, when there is a addition, okay, that is referred to as, now this is a 21st chromosome. So if this 21st chromosome is being duplicated once more, that would result in the formation of Down syndrome. And in the case of Klinefelter syndrome, what happens is it is not an autosomal chromosome that is being increased here. You have an increase in the what? In the X chromosome. And these Klinefelter syndrome is being found mostly in the males, which are having, they have an extra X chromosome. And so that would result into an infertility to them. Okay. Now, this is another case of mosaicism. Okay. I had showed you about, this is about a child. It's a case history which has been published already. It's a tetrasomy, a double tetrasomy, which has been found in two different chromosomes, 8 and 18. And this child has been born with lots of birth defects. Like, okay, he seems to be not like he's having some congenital heart diseases. At the same time, a type of mosaicism has been found on him. He is slow in his growth rate and a little behind than the other children. For everything, he's got different disease symptoms, asthma related, heart related, and all like that defects have been found on this child, which is having who is having a tetrasomy, double tetrasomy. So these are the cases of chromosomal mutations, mainly caused by the aneuploidy in the case of humans. And one reason that we say that as the age of the parents increases, the functional efficiency of the egg as well as the sperm would decrease, and that could further lead to what drawbacks in the after fertilization. So it's always preferred to have a baby at a particular age, which of course in ladies in the case we see that the greater the age goes, the healthiness of the egg it decreases. So I hope this is clear to you. We will continue about the various other chromosomal variations and mutations in the next coming sessions. Thank you.